The chapter is Principle of Inheritance and Variations. This is the genetic chapter from class 12th, biology. I want to discuss in this chapter related reasons with the inheritance. It means we are all you know very well we have similarities or dissimilarities that you call variation is the heredity, passing of the characters from their parents to their offsprings. This branch is known as hereditary branch. And when we study the reasons of the variations that occur in offspring, offsprings are very much different from their parents. So when we study the reasons related with this heredity, this is genetics actually. So what do you think? Genetics means heredity plus reasons of the variations. So some definitions are on the screen. Genetics is the study of inheritance, heredity, and variation of the characters, or study of genes and character. Inheritance means passing of the characters from parents to their progeny. Actually, this is the basis of heredity. So heredity is actually the branch related with the inheritance and variations. Variation, as I told you, the similarities and dissimilarities with their parents. Characters. Character is actually the physical features. You are observing that eye color, for example, height of the individual, for example, height of the plant, color of the flower. This is actually character. But when the height is showing variation, for example, tall height and dwarf, this is actually the trait. Blue eye, brown eye. Red flower, white flower. This is actually trait. So character is a one main physical feature and trait is two types of this feature. Clear, bachcho? Next is the alleles. You know very well the gene. Gene is actually the, today we know the gene. Actually, in his Mendel's time, no one know about the gene. So today it is very clear that a small segment of the DNA that control the character with the help of polypeptide chain formation. That is called gene. So as I told you, height is the character. It means there is a one gene for height. But when you are talking about the trait, there are two traits, tall and dwarf. It means there are two genes for the tall and dwarf. So at this level, now genes are known as alleles. Clear, bachcho? Alleles. It means capital T, small t is the two alleles of the one gene that is height of a plant. This is also known as allelomorph. So clear? Two alleles of a gene for the character height. Clear? Now next is the homozygous term. Homozygous term means when the two alleles are similar. For example, capital T for height, tall, small t for height, dwarf, capital Y, for example, yellow color, small y, for example, green color, whatever, capital R for red color, small r for white color. So when the two alleles are similar in the true breeding means there is no mixing of the characters. Clear? So that is called the homozygous character. As you are seeing two chromosome, homologous chromosome, one is capital B, another is small b. This is a heterozygous. Another homologous chromosome, when capital B, or in a small b. So these two alleles are different. It means homozygous condition. And when you are studying two alleles are different, these are heterozygous condition. Similar homozygous and different heterozygous. So the condition in which chromosome pair carries dissimilar alleles of a gene, that is capital T, small t, capital Y, small y, or inside you are seeing capital B, small b. These are heterozygous character. Actually, this time I'm explaining some related important terms. Those are being used in this chapter. Dominating character means when you will cross two different parent having a pair of character. For example, tall plant cross with the dwarf plant. So in F1 generation means bacho first generation. F1 generation, you will see the character phenotypically. You are seeing physically, observing with your eyes. Yes, this plant is tall, this plant is dwarf. Clear? This is called phenotype, physical expression of a character. So when you are observing the phenotype character, you will see in the F1 generation, there is one character out of two parents. One character is seen in the first generation. Another character is hidden. So one character that is seen, shown in the F1 generation, this is called dominant character. And another character that was hidden or masked in F1 generation. This character is known as recessive characters. Clear, bachcho? So with the help of example, you can understand these terms more clearly. Now next, as I told you, phenotype, physical expression of a character. Genotype means, as you have seen, capital T, capital T, capital T, small t, है ना? अभी आपने इसमें देखा ना, एलियस वाले टर्म में देखिए, कैपिटल टी, स्मॉल टी, है ना, स्मॉल y, capital Y. So these are actually genetic makeup. Genetic makeup. So genetic constitution of a character, this is called genotype. And when the two parents are crossed, an individual showing the character of both parents, one may be similar or may be dissimilar. Clear? When the unlike characters, an individual produced by mating of genetically unlike parents, and the individual offspring is showing both the character, this is known as hybrid. Clear? So after understanding some terms, now come to the main point related with the Mendel's law of inheritance. Actually, the first time how characters are passing from parents to their offspring, it was studied by the Mendel. He was not actually a biologist, but he developed some interest towards this biology really part, that is how characters are passing, how tall plant produce the dwarf plant also, and how the red flower plant showing the white flower also in few generation. How? So he developed interest. And then he studied 7 years, 1856 to 1863, on this garden pea. The name of garden pea, Pisum sativum. Botanical name is the Pisum sativum. So he conducted some hybridization experiment on this Pisum sativum for 7 years. And then he found that how characters are passing, something that is passed from parents to their offspring. He said something. He didn't know about the genes. We know very much about the genes, but that time gene concept was missing. So he said something, and after some time, he said something were to factors. So these factors are responsible for the character. So for this, he studied 7 varieties of a Pisum sativum pea plant. And then he concluded that what is the actually inheritance, heredity, and what is the actually base of the genetics. So when he studied 7 varieties, it means he crossed with desirable characters. Pisum sativum, for example, red color flower, a violet, you can say violet color flower and white color flower. So when you crossed with your desirable characters, this process is known as hybridization process. And you are controlling the pollination because you don't want to contamination of the violet color flower. This is actually Pisum sativum, pentamerous flower, Papilionaceae family member, vexillary petals. You know? So in this case, in one flower, removal of the stamens, and another flower, removal of the female part, that is gynoecium. So removal of the anthers, that is actually a step known as emasculation process. So when you want to cross desirable characters in plants, first you have to select 2 desirable variety of the plant. So in this screen, you are seeing Mendel did selection of 2 pea plant with contrasting character, violet color and white color flower, tall and dwarf plant. Clear? And then removal of the anthers from one plant to avoid self-pollination. This is female flower emasculation process. Obviously, this will be occur in bisexual flower. So in bisexual, you want to one character as a female or another character as a male. So you have to remove anthers from the flower. This process known as emasculation. And then you will dust desirable pollen grains from the male plant on the female plant. So this transferring of the pollen grains from male to female, this is known as pollination. And after pollination, F1 generation and further F2 generation was collected as a seeds to produce new offspring. Clear? So these are few steps related with the Mendel's work. As I told you, Mendel studied 7 true breeding varieties. True breeding means the individual showing only one character at a time, only one trait at a time, not a mixing of character. In next generation, in successive generation, you will observe only one type of character. So this is known as true breeding plants. So you can observe on this scheme, there are 7 characters, stem height. First, in F1 generation, appear character is the dominating character, as I told you, hidden character is the recessive. So when he crossed tall and dwarf plant, on the right hand side, you will see diagrams also related with the tall flower and tall plant and dwarf plant, violet and white flower, full constricted pods. Clear? So these are the same stem height, for example, tall and dwarf, flower position, axial and terminal, flower color, violet and white, pod shape, inflated and constricted, pod color, green and yellow, seed shape, round and wrinkle, and seed color, yellow and green. So Imagine yellow color seed are actually dominating color. Generally, we know, yes, greens matter is very tasty or very desirable. But actually, this green is the recessive character in the seed color. But green color of the pod is dominating character. I hope clear these 7 true breeding varieties selected by the Mendel. And after selection, true breeding, he only concentrate on the first character at a time, first contrasting trait on it at a time, not haphazard. किसी का tall ले लिया तो किसी का yellow भी ले लिया, seed ले लिया, किसी का shape ले, no, no, no. He only focused at a time only one contrasting character. So this is known as inheritance of one gene. According to them, passing of one factor. That's showing 2 alternate alleles. So this monohybrid cross means a cross involving 2 plants differing in one character pair. देखिए screen पे, tall and dwarf plant, cross between tall and plant. Yellow and green, cross between yellow seed plant and green seeded plant. So Mendel crossed tall and dwarf pea plants to study the inheritance of one gene. This is known as monohybrid cross. Inheritance of one gene means passing of the one allele in one gamete. Parents tall, homozygous plant, homozygous tall character, it means true breeding for tall character. And another plant, this is the dwarf plant, this is true breeding plant, small t, this is a genetic makeup, capital T for the homozygous plant. At the time of gamete formation, you are observing in one gamete, one allele is passed, in another gamete, another allele is passed. So this is gamete formation. Same type of gametes in the tall plant, same type of small t gamete in the dwarf plant. And these are crossed as a male or female. F1 generation was appeared as a tall plant. But genetic makeup, when studied, it is found that this is the heterozygous tall plant. Heterozygous, as I told you, 2 different allele of one character, capital T and small t. Actually, that time it was not known about the genetic makeup. Actually, Mendel is simply growing these pea plant tall and dwarf and crossed in the F1 generation. He observed in field, yes, all are tall. Very good, all are tall. But second question was, what about dwarf character? So he curious and he crossed self-crossing. F1 generation selfing means all the tall plants of the first generation are self-crossed. Then he found in F2 generation, dwarf character appeared. You are seeing it on the screen. Dwarf character appeared. He amazed. How possible? In F1 generation, it was not shown, but in the F2 generation, it is shown. Then, actually, there was no physical explanation of this alleles or genes that time. So number of the biologists not considered these experiment, not accept these results actually. This was the reason. But now, after knowing number of the other characters plants crossed and found the same result that Mendel already given. Now you are saying that Mendel is the father of genetics only because of that He worked hard. He gave number of the experiment. For one result, about 29,000 experiment he performed. And then gave the final result of X1 character. So now you know very well, because we know about the genetic makeup. That time was not no nucleus, chromosome, because there was no microscope. But now it is very much clear. In F1 generation, heterozygous, one character is dominating, another character is hidden. But in F2 generation, when self-crossed, due to the presence of both the alleles, this hidden character as a recessive parent shown. Clear? As a recessive parent is shown. So in this, you are seeing in F1 generation, heterozygous condition, capital T, small t, when these are crossed, self-crossed, and then found out of 4, 3:1, 3 tall and 1 is the dwarf recessive parent type. So this is called monohybrid phenotypic cross. But when studied the genetic makeup about this, 1:2:1 ratio of this monohybrid cross, because 1 is the pure tall. Always give successive generation, you will get only tall plant, not dwarf plant, because there is no allele for the dwarf character. And these 2 plants out of 4, these are again heterozygous. So you will get tall and dwarf plants also, because there are both the alleles, capital T, small t, present. And 1 is the only dwarf pure parent type offspring. So in successive generation, it will only produce dwarf plant, not tall, because there is no alleles of the tall character. Clear, bachcho? So that is monohybrid genetic. Your genotype ratio is homozygous tall, 1:2:1 dwarf. Clear? I hope clear this point. So finally, you can say Mendel made similar observation for the other pairs of trait. As I told you, 7 characters. Same result he observed. He proposed that some factors, now something is replaced by factors finally. Some factor that you call today as a gene. Some factors were inherited from parent to offspring. Factors are now called gene. So what is a gene? Gene is actually a small segment of DNA, and DNA form the protein, particular protein for any character. Clear? So genes are actually the responsible for the character, from passing character from parents to their offspring. Clear, bachcho? So on the basis of this, Mendel proposed a law of dominance. First law. Actually, Mendel gave the 3 laws: law of dominance, first law of segregation, second law, and third law is law of independence, assortment. This is related with the dihybrid cross. So today in this lecture, I am only focusing on the monohybrid cross. So on the basis of, you have studied, on the basis of this monohybrid cross, first law given by the Mendel, law of dominance. As I told you, when 2 contrasting parents character crossed, when these 2 alleles are present in the F1 generation as a genetic makeup, but phenotypically you are observing only 1 character in F1 generation, that is tall. So this is known as law of dominance. So characters are controlled by discrete units called factors. factors occur in pairs, capital T, small t, capital T, capital T. I mean to say, factors occurs in pair. In a dissimilar pair of factors, yani capital T, small t, one member of the pair is dominates, that is dominating character, and other is hidden or masked, that is called recessive character. So, out of two alleles dissimilarities, one is expressed and another one is hidden. Hidden is recessive and expressed is dominating character. So on the basis of this, law is known as law of dominance. And this second law is also very important, law of segregation. As I told you, at the time of gamete formation, capital T, small t gets separated. You know? See, capital T is one factor and small t is the another factor. So it was only gets segregate at the time of gamete formation. Capital T will form same type of similar gametes T and T. Similar pair of factors homozygous for dwarf small t, but heterozygous members dissimilar pairs of factors heterozygous segregate as a T, capital T, as a small t dissimilar gamete. So law of segregation means although in F1 generation small t is hidden in the presence of capital T, but it is not blended, it is not completely destroyed. It was only hidden or masked in the presence of dominating allele. But in F2 generation, when it is segregated at the time of gamete formation, it is independently segregated. So when, as I told you, 1:2:1, में आपने देखा small t, in homozygous condition, express its parent type character as a dwarf. So it means at the time of gamete formation, this is segregated. It is not destroyed its product. Products mean expression, I mean to say. Clear? Assalamualaikum. As in first lecture, I told you about the law of dominance. Clear? Mendel's law. Mendel's have given two laws related with the monohybrid cross. First law of dominance and second law of segregation. So let's start with the law of segregation. Law of segregation means at the time of gamete formation, two alleles get segregated. For example, capital T, these are the homozygous individual. At the time of gamete segregate as a capital T, two different. So during gamete formation, the factors or alleles of a character pair present in parents segregate from each other such that a gamete receive only one of the two factors. As you know very well about the meiosis, during meiosis, you know, at the time of anaphase 1 stage, the chromosome become half. So that is the same thing. Actually, if you have clear concept of the meiosis, then you can understand the law of segregation. So law of segregation is universal law. There is no exception, no exceptional case related to this law. Any individual, either homozygous or heterozygous, at the time of gamete formation, two alleles have to get segregated. Homozygous parents produce will similar gametes, capital T for example, small t for, and heterozygous parents produce two kinds of gametes, each having one allele with equal proportion. As you are seeing on the screen, dissimilar gametes of the heterozygous capital T and small t. So any type of individual, in any plant, in any animal, in any kind of offspring, law of segregated is accepted. at the time of gamete formation, alleles have to get segregated. Whenever fertilization occurs, male and female gamete fuse, again it becomes 2 in number. Clear? That is the point. And now come to the back cross and test cross. What does it mean? Actually, when you are observing in the N1 F1 generation, all plants were tall. So how can you understand this is the hybrid tall or heterozygous tall? Or you can say there are two alleles, one is the tall allele and one is the dwarf allele. Or this is a homozygous tall. It means both the alleles are similar. So to know about this, F1 individuals are homozygous or heterozygous, you have to cross back. So back cross means cross between a hybrid, I mean to say F1 generation, maybe F2 generation, whatever, hybrid, and its any parents. So when you will cross the F1 hybrid with the dominating true breeding parents, you will get all this same type of individual. I mean to say, for example, if I want to say F1 tall individuals are crossed with the homozygous true breeding tall parents, you just do it with the help of pencil on your copy. Hybrid capital T small t cross with the tall parents, capital T, capital T. You will get next generation all are tall. Maybe hybrid or maybe true, but all are true. So there is no testing of F1 offsprings as heterozygous or homozygous. This is called outcross. Actually, backcross two type. One is the outcross. When you hybrid parent cross with the dominating true parent, outcross. But when you will, as screen showing, but when you will hybrid tall cross with the recessive parent dwarf, you will get 50% tall, although this is a heterozygous. In next generation, you will be able to get dwarf and tall, both the plants. But this is tall, dominating. All individuals 50% tall. And other 50% are pure dwarf parents. So homozygous test, monohybrid test cross ratio is 1:1 or 50% tall or 50% tall. 50% dwarfs are true breeding, dwarf plants, just like parents, recessive parents, and tall plants are hybrid with recessive allele. So you can say heterozygous. Actually, this is the crossing of an organism with dominant phenotype to a recessive individual. So you'll get test cross ratio 1:1. This is the NCERT diagram you will see in your book. Test cross is used to find out the unknown genotype of a character. Already told you, test cross is actually for testing. F1 generation individuals were heterozygous, homozygous. This is the actually want to test. See in this diagram, on the left hand side, you are seeing homozygous recessive, white, two types of gametes form, small w, small w. And in middle, you are seeing dominant phenotype, violet color, but genotype is unknown. For example, when this is crossed, homozygous recessive and This is the violet color dominating phenotype crossed. All flowers are violet. All flowers. So unknown flower is homozygous dominant. Interpretation of this experiment. But in another cross, when the homozygous recessive crossed with the violet color flower and found that half of the flowers, it means 50%, as I told you, are violet and 50% flowers are white. So now it's clear unknown flower is heterozygous. In homozygous recessive, you are seeing one type of gametes form, small w, small w. And in the dominating phenotype, in one's left-hand side, one type of gamete, capital W form. And right-hand side diagram is capital W, small w gametes are formed. Actually, when this back cross is done with the dominating parent, scientists were unable to understand, yes, this is the heterozygous or homozygous. But when it is crossed with the homozygous recessive parent, F1 generation, because F1 generation was heterozygous, not homozygous. That's why able to get another recessive parent character in the next generation. So you can say test cross ratio is 1:1 in F2 generation. Mendel conducted test cross to determine the F2 genotype. Clear, bachcho? So that time Mendel was unknown, not recognized his work, because biologists not accepted that mathematical statistical analysis was given in these experiments. Communication was not there that time, as now very fast communication. So work of the Mendel was not publicized that time. And third reason was unrecognition is that time. Not the physical appearance of any alleles. What is this actually allele? But after no microscope, no nucleus knowledge, no chromosome knowledge. So his work was unrecognized. But in 1900, number of the scientists who worked on the number of the plants and animals and found that same result that was Mendel published. Then his importance was recognized. And now he is known as father of genetics. So I hope clear. Cross, test cross, as well as back cross, law of dominance, and law of segregation. Thank you.